Hey, and welcome back to my forestry lecture series. This is the third video and it will be focusing on roots. This is also probably the last forestry video since the season is coming to an end. Here's my contact info in case you have any questions or comments. Some things you should be familiar with for this video, either from previous videos or from background knowledge, are the types of tissues found in trees, which include dermal tissue, ground tissue, and vascular tissue in particular, including xylem and phloem and their functions. And the two clades that make up angiosperms, which are monocots and dicots. Just basically know that they exist because the internal root structure will differ between them, just like leaf structure. Hopefully you are familiar with what an organ system is, just a group of organs working together to accomplish tasks like the muscular or skeletal system in humans. The organs of plants, including trees, are typically divided into two organ systems: the shoot system, which is typically all above ground, and the root system, which is typically all below ground and is the focus of this video. The root system includes roots, and the shoot system includes everything else: leaves, buds, stem, trunk, flowers, etc. The normal root to shoot ratio for a tree is around 1/5 to 1/6, meaning the dry weight of the shoot system is normally 5 to 6 times heavier than the weight of the root system. The ratio stays relatively constant to ensure neither the root system or the shoot system grows out of proportion to the other system. A root is an organ in plants that exhibits geotropic growth, which means it grows downwards, as opposed to the anisotropic growth of a tree stem, which means it grows upwards. Roots have many different functions and are often specialized to be more efficient at a certain task. Pretty much all roots carry out these four functions. They anchor and support trees so they don't just fall over. They absorb and support water and nutrients around the tree. They act as storage for food and nutrients, and they produce certain hormones that promote growth in the shoots system. Some roots are also specialized to perform specific functions, such as storing food, performing photosynthesis, or even reproduction of the tree. Now we're going to discuss the difference between the two types of growth: primary and secondary. Primary growth is the kind of growth that increases the length of the shoots or roots of a tree. Pretty much all of the growth discussed in this video will be primary growth. Secondary growth, however, is an increase in thickness of the shoots or roots. For the most part, only dicots and gymnosperms exhibit secondary growth, not monocots. Secondary growth was not included in this lecture since it is more appropriate to learn while studying trunk growth, not root growth. One of the most important ideas in this lecture is the concept of meristem. Meristem is a type of plant tissue that is neither ground, nor dermal, nor vascular. It is undifferentiated, meaning the cells have not been specialized yet. The cells have the potential to differentiate into any one of the three tissue types, but are currently none of them. Meristem cells are constantly undergoing cell division through the process of mitosis. This mitotic cell division is responsible for all of the growth done by a plant, as these newly produced cells could soon become any and every different type of tissue within the plant. There are three main types of meristem found in plants, but there is only one type that is relevant to this lecture: the apical meristem, which is found at the tips of roots and shoots. Apical meristem is responsible for all primary growth, which again is any increase in length of a plant. Pretty much all sources divide roots into two different systems: the taproot system, which is found in most dicots, and the fibrous root system, which is found in most monocots. Many plants can have a combination of these two systems. The taproot system has a large root called a taproot that is bigger than all the other roots, and as it grows downwards, lateral or horizontal roots begin to grow out of it. The fibrous root system is composed of a big mass of small roots with a similar diameter that is concentrated in about the first foot of soil. These two root systems only tell half the story, however, because as trees age, their original root systems become less obvious and they will eventually develop into a set of more mature root systems that will be covered in the main coat of this video. In a typical root cross-section, you will see three regions of cells: the root epidermis, the cortex, and the stele. The stele is the center of the root and is where vascular tissue in roots is located. Roots are exarch, meaning that the vascular tissue develops from the outside to the center. This contrasts the pattern of development of stems, which are endarch, meaning they develop from the center outwards. Protoxylem and metaxylem are the two components of primary xylem, or xylem produced by primary growth. Protoxylem is produced first and matures earlier than metaxylem, but protoxylem cells are smaller and less efficient at conducting water. There is also protofloem and metafloem, which mirror the properties of their xylem counterparts. You will find protoxylem on the outer side of metaxylem because roots are exarch. Protoxylem develops first because it's closer to the outside of the root. It follows that since stems are endarch, the metaxylem is on the outer side of the protoxylem in stems. The root tip can be divided into four main parts. Starting from the tip of the root is the root cap, which protects the root as it grows through the rough and abrasive soil. The root cap covers the region of cell division, which is where all the new cells of roots are produced during primary growth. Above the region of cell division is the region of elongation, which is where the cells produced in the region of cell division undergo growth and development, increasing in size and becoming more specialized. Above the region of elongation is the region of maturation, also called the root hair zone. This is where cells become almost or fully developed and where the growth of root hairs takes place. This makes the four parts of the root tip, the root cap, the region of cell division, the region of elongation, and the region of maturation. Nearly all trees form symbiotic relationships with fungi in the soil. These symbiotic associations or relationships are called mycorrhizae, or singular mycorrhiza. Some trees also form symbiotic relationships with bacteria in the soil that are capable of performing nitrogen fixing, or converting unusable atmospheric nitrogen into nitrogen compounds that trees can use. These bacteria often form growths on the roots called root nodules, which contain large amounts of nitrogen-fixing bacteria. Now we can go into a bit more detail for each of the main root functions. They anchor and support trees so that they don't fall over as they become heavier, or during storms or floods or any other harmful weather events. They absorb and transport water and nutrients such as nitrogen, phosphorus, and potassium found in the soil. The majority of this absorption occurs near the smaller, newly grown roots, similar to how in the circulatory system of the human body, where the exchange of gases takes place in the smaller capillaries rather than larger arteries and veins. The cylindrical shape of roots has a high surface area to volume ratio, which allows water and nutrients to be absorbed more efficiently. Roots also have several internal barriers that prevent water loss after it has been absorbed, so there's no concern about excess water loss arising from a large surface area like there is in leaves. In the center of the roots lies xylem and phloem, which are responsible for transporting water, nutrients, and sugars produced by photosynthesis. Xylem transports the water and nutrients solely upwards, whereas phloem transports sugars to all parts of the tree to fuel cell growth, development, and metabolic activity. Roots also act as a storage for the sugars made from photosynthesis and for water. Larger roots are able to store more carbohydrates or sugars. These root storages are important for tree regrowth in the spring when it is impossible for deciduous trees to perform photosynthesis due to a lack of leaves. The energy required to grow new leaves therefore comes from the root storages of carbohydrates. Carbs are also stored in the stem of trees, but oftentimes storing them in the roots is more favorable because they are more protected from animals and are in a much more stable environment. Roots also produce hormones, most importantly cytokinin and gibberellin, which promote the growth and development of the shoot system. It is advantageous for roots to control the growth of the shoot system because it helps maintain a constant root-to-shoot ratio. If the roots are growing a lot, they will produce more hormones, which will promote more growth in the shoot system, ensuring that the roots don't get too big relative to the shoots. There are additional functions that only certain types of specialized roots can do. I won't go over all the types in this video because I don't feel like it. Some examples include photosynthesis. Some roots that go above ground are capable of performing photosynthesis. Movement. Contractile roots are specialized to contract, pulling a plant downward into a more stable environment. Aeration. Plants in watery environments have roots called pneumatophores, which allow gas exchange to occur in the roots that would have been prevented from the watery soil. Yes, roots also need to carry out gas exchange, not for photosynthesis, but for metabolic activity. You will often see trees with many different types of specialized roots all working together. The association that most dicots have taproot systems and most monocots have fibrous root systems is a bit misleading. This is stated in the context of all angiosperms, or all flowering plants, including many plants that are not trees. If we only consider trees, the two root systems discussed, taproot and fibrous root, are relevant only when describing the roots of a young tree. And young trees may have taproot systems or fibrous root systems, regardless of whether they are a monocot, dicot, or gymnosperm. Each tree eventually develops one of three mature tree root systems. First, let's talk more about the roots of a young tree. When a tree seed begins to sprout or germinate, the radicle is the first thing to come out, also called first root or embryonic root. If a young tree uses a taproot system, the taproot will develop from this radicle. The taproot will grow further and further into the soil, and horizontally oriented roots will eventually begin to emerge from this taproot, producing a taproot system. Some examples of trees that employ this young root system are most oaks, pine, walnut, and hickory. Many young trees with a taproot system will grow until the taproot reaches an obstacle and then switch to a fibrous root system. If a young tree uses a fibrous root system, the radicle dies during or immediately after seed germination, and small roots begin to sprout at the base of the dead radicle. As the tree begins to age, roots start growing from the stem. Roots that do not originate from a pre-existing root are also known as adventitious roots, such as the roots that grow from the stem. These adventitious roots eventually reach the ground and begin to grow and integrate into the roots already present, producing a fibrous root system. It is important to note that adventitious roots can develop in both monocots and dicots. Some examples of trees that employ this young root system are maples, ashes, and cottonwoods. Now that the initial development of tree root systems is hopefully more clear, we can discuss the three root systems seen in mature trees. The first type of mature root system is called a lateral root system, a flat root system, or a plate root system, and is the most common of the three, being present in around 80% of tree species. This root system is dominated by lateral roots, also called horizontal roots. These roots grow horizontally and are typically close to the surface of the soil. Because the roots are spread so widely, this root system is able to effectively anchor trees while having less mass than other root systems. Some examples of tree species that use this system are ash, cottonwood, hackberry, maple, fir, spruce, and beech. If a lateral root system has many sinker roots, or roots that originate from lateral roots and grow vertically downward, the system may be called a sinker root system. The second type of mature root system is called a heart root system or an oblique root system, and it is characterized by the presence of many heart or oblique roots. These roots are similar in structure to lateral roots, but grow diagonally outwards. Heart root systems often also include lateral roots and sinker roots, forming a particularly compact root structure. These root systems depend on the resistance of soil to anchor the tree, making them particularly vulnerable to tipping in wet soil. Some examples of tree species with this root system are birch, larch, and some oaks. In almost every species of tree that begins with a taproot, the taproot is gradually relied on less and less as more roots develop. The taproot begins to shrink and most die in a root away after the seedling stage. However, around 5% of trees have the third type of mature root system, the taproot system, in which the taproot remains the most important root in the root system throughout the tree's lifespan. This mature root system is also called the taproot system since it is a continuation of the taproot system seen in young trees. Taproot systems also possess lateral and oblique roots, but to a lesser extent than the previous two systems. Examples of tree species that use this root system are some oaks and pines, hickory, sweet gum, tupelo, and walnuts. After going through these root systems, I want to emphasize that nature is rarely predictable. Trees may have exactly what one of these root systems describe, or they may have a mix of two or all three. These are just the observed trends of root growth in trees, and trends are not representative of every single tree you might come across. Additionally, root growth is opportunistic, meaning it will only grow where the soil can sustain it. In other words, where conditions are favorable. All trees are capable of forming deep root systems, but not all trees develop them due to the less favorable conditions in deeper soil. The majority of roots are found in the top 6 to 18 inches of soil because of the abundant water and nutrients found in this region, along with the fact that obstacles are less likely to occur here when compared to deeper soil regions. While genetics do play a role in determining the root system a tree develops, due to the opportunistic growth of roots, the soil conditions are typically more important in determining the root system. Lastly, tree root systems notably help prevent soil erosion by holding on to the soil it grows on. Now we'll take a look at the cross-section of a developed root that has not yet undergone secondary growth. Since the development of roots is exarch, meaning they develop from the outside in, we will build our root in the same manner. The root epidermis, also called rhizodermis or epiblemma, is the outer single layer of cells in the root cross-section. The purpose of the root epidermis is to absorb water and nutrients from the soil. It is made of barrel-shaped parenchyma cells with characteristically thin walls, which also make absorption easier. The root epidermis also has root hairs, also called root trichomes or rhizoids, which aid in the absorption of water by increasing the absorptive surface area. It is important to note that root hairs are not separate structures from the epidermis. They are simply tubular extensions of already existing root epidermis cells. Unless it is an aerial root, root epidermis does not have stomata, and it will have an extremely thin or absent cuticle since their task is to absorb water, and a waxy cuticle can make this hard. Additionally, aerial roots may have an epidermis that is several cells thick, called a multiple epidermis or velamen, which often have the ability to store water from humid atmospheres. The next tissue in the root cross-section is the cortex, which has two main responsibilities: to transport the water and nutrients absorbed in the root epidermis to the vascular tissue, and to store reserve food for the tree. There are two basic types of cortical tissue: solid and lacunnate. Solid cortex is made of compact parenchyma tissue with very small intercellular spaces. This type of cortex is common in trees that grow in well-drained soil. Lacunnate cortex is a little more complex. It has an outer and inner layer of compact parenchyma cells, but in between these layers there are long plates of cells with large intercellular air spaces between them, forming air cavities called lacunae. This type of cortex is common in trees that have roots which are periodically waterlogged or entirely immersed in water. Remember that the main function of the cortex is to transport water and nutrients absorbed in the epidermis to the vascular tissue in the center of the root. The transport of water containing nutrients through the cortex may be apoplastic or symplastic. Apoplastic transport moves water in between the cell walls of the cortex and does not require energy. Symplastic transport moves water through the cortex cells themselves, which does require energy. As you can see from the diagram, apoplastic transport follows the blue arrow and symplastic transport follows the red arrow. The innermost layer of cells of the cortex is called the endodermis, which is only found in roots. The purpose of the endodermis is to regulate what goes into the tree's vascular system. After root hairs die on a section of a root, the cortex and root epidermis also die and are shed, leaving the endodermis as the surface of the root until bark forms around it. The radial walls of endodermis cells are thickened with suberin, forming Casparian strips on these walls. Casparian strips form impermeable, waterproof walls that prevent apoplastic transport, or transport in between cell walls, from occurring, making symplastic transport the only option. When the absorbed water and nutrients travel through the endodermis cells themselves, the types of minerals and the amount of water entering the vascular tissue can be regulated. Older endodermis cells may have lignin and more suberin be applied to all cell wall surfaces, forming a waterproof barrier. Water and nutrients are still able to transport through the endodermis after it has become waterproof due to the existence of passage cells. Passage cells exist every so often in the endodermis and do not have suberin and lignin applied on all cell wall surfaces. They keep thin transverse walls and just have Casparian strips on radial cell walls, allowing the symplastic transport of water and nutrients into the vascular tissue. When endodermis cells pump water into the vascular tissue and don't let any out, root pressure is created. Since water cannot go back out of the endodermis, as more water is added, the only way for excess water to go is up, where there is less root pressure. The upwards direction will always have less root pressure because water is continually being used up by the leaves through processes like transpiration and photosynthesis. Root pressure is one of the factors that allow xylem to transport water upwards without using energy, in addition to the cohesion tension theory mentioned in the leaf lecture. Root pressure is maintained by older endodermis become almost completely waterproof, with the exception of passage cells as described above. A phenomenon called guttation can occur when there is not enough relief of root pressure. For example, during the night, the rate of transpiration is lower and photosynthesis is not occurring from lack of sunlight, so less water is being used up. Since the roots are still absorbing water during the night, there is no place for the excess water and nutrients to go except out of the plant, which is what guttation is, the leaking of xylem sap, a combination of water and nutrients, out of the edges of leaves. Guttation usually occurs when the soil of a plant or tree is overwatered, causing a buildup of root pressure. The stele or vascular cylinder of a root is everything inside the endodermis, containing the primary xylem, the primary phloem, the conjunctive tissue, and the pericycle. The pericycle is the outermost covering of the stele, and it is only a single layer of parenchyma cells. Inside the pericycle is conjunctive tissue, which is made up of loosely arranged parenchyma cells specialized for water storage. Embedded within the conjunctive tissue is the primary vascular tissue. Remember, it is primary because it resulted from growth in root length, not from the growth in root thickness. The arrangement of this vascular tissue depends on whether the tree is a monocot or a dicot. The more commonly studied root is that of a dicot, which typically has between 2 and 6 strands of xylem. Since the development of vascular tissue in tree roots is exarch, the metaxylem of each strand is located towards the center and forms a sort of core of metaxylem, and the protoxylem arms branch off of that core. The stele can be classified based on how many arms of protoxylem it has: 2 for diarch, 3 for triarch, 4 for tetrarch, and so on. A root is referred to as protostelic if the xylem forms a solid core with arms or lobes, which is typically the case in dicots. Outside the xylem is the phloem, arranged in between the protoxylem strands, and the number of phloem strands will always equal the number of xylem strands. The outer-facing cells of each of these phloem strands are called protophloem, and the inner-facing cells are called metaphloem. In contrast to this, monocot root steles will always have more than 6 strands of xylem, which can be described as polyarch. These strands, along with strands of phloem, are radially arranged around a central parenchymatous pith that is not present in dicot roots. A root is referred to as siphonostellic when vascular tissue surrounds a pith, which is typically the case in monocots. Here is a finished diagram of a mature root cross-section. It displays the two types of cortex and the two types of monocot stele. It is important to note that just because there is a lacunate cortex does not also mean that the stele will be monocot. It is just a way of displaying both types of cortexes and steles. As stated before, the tip of the root is typically divided into four main parts. Just like in the leaf lecture, we'll build the root tip one part at a time. It is important to note that the boundaries between the three upper regions of the root tip are not abrupt. They gradually transition between one another. They are separated on the activity the majority of the cells in each region go through. Additionally, for a sense of scale, the four root parts mentioned below all occur within the first centimeter of the root tip. If you remember from this video's primer, meristem cells are undifferentiated and constantly dividing through the process of mitosis. Within the region of cell division, root apical meristem, or RAM, mitotically divides to produce all of the new cells involved in primary growth of a root. However, in the center of the RAM, there is a region of cells that divide slowly or not at all called the quiescent center. These cells are capable of resuming meristematic cell division if the outer layers of RAM are damaged, allowing the RAM to recover. As you move up the region of cell division, the apical meristem begins to differentiate into three primary meristems: protoderm, ground meristem, and procambium. Each of these primary meristems will continue to differentiate into different tissues as you move further up into different regions of the root tip. The protoderm will differentiate into the root epidermis, the ground meristem will differentiate into the root cortex, and the procambium will differentiate into the tissue found in the root stele, including the primary vascular tissue and pericycle. Above the region of cell division, we can add the region of elongation. This region is fairly self-explanatory. It is where cells produced in the RAM elongate and expand greatly. Pretty much all of the increase in length a root experiences occurs here. Although cells are still differentiating, none of the cells found in this region will be mature. The protoderm, ground meristem, and procambium are still present. This region is also called the absorption zone or root hair zone because it is where root hairs grow, and the beginning of this region is sometimes defined as where root hairs are first found. Root hairs are simply extensions of epidermal cells, not separate cells, which serve to increase the absorptive surface area of the roots. They are ephemeral or short-lived and only last about 5 days before dying. It is in this region of maturation where the primary meristems differentiate into distinctive mature tissues such as root epidermis, root cortex, endodermis, pericycle, and primary vascular tissue. The cortex cells also begin to fulfill their duty of transferring water and nutrients from the root epidermis to the vascular tissue through the endodermis. Lastly, we can cap off the root tip by adding the root cap, which is on the very tip of the root. The main function of the root cap is to protect the vulnerable cells of the RAM behind it. Since it is the tip of the root, it is constantly pushing past abrasive soil particles that erode away the bottommost layer of cells exposed to the soil. In fact, only 4 to 5 days pass between a root cap cell being produced and it being eroded completely away. In order for the root cap to not erode away entirely, it needs to constantly undergo cell division. It accomplishes this by having the uppermost layer of cells of the root cap be meristematic. In monocots, there is a meristem separate from the root cap but responsible for all root cap growth called the calyptrogen. Cells produced from this root cap meristem are then pushed downwards in rows towards the bottom edge of the root cap, shown as group A in this image. As they are pushed downwards, they begin to differentiate into root cap parenchyma. And remember, once cells differentiate, they are no longer meristematic. The cells on the edges of these rows divide and expand outwards, forming the side regions of the root cap, which is depicted as group B in this image. Root cap parenchyma is characterized by having the ability to secrete mucigel, a slimy substance that lubricates the growth of a root through the soil and also allows roots to absorb nutrients more easily. The root cap is also responsible for a root's ability to detect gravity and its geotropic growth. Root cap cells contain amyloplasts, which are little structures inside cells that contain starch. These starch-filled amyloplasts then settle on the bottom of cells, which triggers the downward or geotropic growth of roots. The region of maturation is the last region in the root tip, so above that is what can be considered mature root. Cells that have not yet finished differentiation in the region of maturation do so here. Suberin and lignin is applied to cell faces of the endodermis, sometimes resulting in passage cells. Continued maturation of the endodermis results in the formation of a watertight sheath around the vascular tissue, which helps keep the water inside vascular tissue for older roots. As previously mentioned, after root hairs die and are shed off, the root epidermis and outer cortex may die and shed off, leaving the endodermis to be the surface of a root until secondary growth occurs and root bark forms. Above the root tip is also where lateral roots originate. The growth of a lateral root is initiated by cell divisions in the pericycle, where cells resume meristematic activity and form a new ram which pushes laterally outwards. When the lateral root emerges from the parent root, it has formed a root cap and vascular tissue is beginning to differentiate, which will eventually connect to the vascular tissue in the parent root. A mycorrhiza is a symbiotic relationship between the roots of around 80% of seed plants and soil fungi. A symbiotic relationship, or a symbiosis, is a long-lasting biological relationship between organisms of different species, and a mycorrhiza is a type of symbiosis called a mutualistic relationship, meaning both organisms involved benefit from the relationship. There are two main types of mycorrhiza, the first of which is called an ectomycorrhiza. In this relationship, the slender fungal cells called hyphae penetrate in between the outer root cortex cells but never go inside the cells themselves. This relationship occurs in the majority of trees, including most conifers and some angiosperms such as oaks, hickories, and birches. The second type is called an endomycorrhiza, and it occurs on a larger variety of plants than ectomycorrhiza, but on a smaller variety of trees. In this relationship, hyphae penetrate through cortex cells and reach deeper into the root cortex, but notably are unable to pass through the Casparian strip of the endodermis. One way to visualize the difference between ectomycorrhiza and endomycorrhiza is to compare them to the methods of cortical transport we covered earlier in the lecture. Ectomycorrhizal fungal hyphae pass in between cell walls just like apoplastic transport, and endomycorrhizal fungal hyphae pass through cells just like the symplastic transport. Remember that a mycorrhiza is a mutualistic relationship that helps both organisms. The fungi in mycorrhiza benefit by receiving food and amino acids produced by the tree, allowing the fungi to grow hyphae outwards into the surrounding soil. In return, these newly grown fungal hyphae dramatically increase a root system's ability to absorb water and nutrients by transporting anything they find into the cortex of the roots. In particular, mycorrhizae are noted for how they significantly increase phosphorus intake of trees. Similar to mycorrhizae, there is another type of symbiotic relationship that can sometimes occur in the roots of trees. It results from nitrogen in the soil often being a limiting factor for tree growth, or in other words, trees have everything else they need to grow except for nitrogen, so they can't grow unless they get nitrogen. Some of you may know that nitrogen is actually the most common element in the atmosphere, so how can it be scarce? Well, trees are unable to break down the nitrogen in the air because the N2 molecule is too hard to break down. That's where bacteria comes in. Some bacteria can break down and use the N2 molecules, and when the bacteria die, tree roots are free to absorb the now usable nitrogen compounds left over from the bacterial death. The process of converting atmospheric nitrogen into usable compounds is called nitrogen fixation. Bacteria of the Rhizobium genus in particular are known for forming symbiotic relationships with trees. Bacteria in the soil are able to enter root hairs through a thin structure called an infection thread, which reaches into the cortex of the root. Cortex cells next to the infection thread divide rapidly to form a lump called a root nodule, and eventually the bacteria fills the cortex cells with bacterial cells called bacterioids, which are capable of nitrogen fixation. This symbiotic relationship can be called a mutualistic relationship in which both organisms benefit because the tree receives usable nitrogen from the bacteria and the bacteria uses energy from the root cells to reproduce within the root nodules. We have now finished the main coat of this video. If you're taking notes, pause the video and take a few minutes to try and write down the topics you remember from the main coat without looking at your notes. If you aren't taking notes, you can still pause this video and try to mentally recite the topics you remember. When you're finished, unpause the video and we'll quickly go over the important topics covered. Make sure to pay extra attention to any that you missed. You can go ahead and pause now. Now let's get to the review. We talked about the root-shoot systems of a tree and how the typical root-to-shoot ratio is around 1/5 to 1/6. Remember that this ratio is maintained because the root system controls the growth of the shoot system by releasing hormones. So the larger the system gets, the more hormones are sent to the shoot system, causing the shoot system to grow larger as well. We talked about the difference between primary and secondary growth, where primary growth is increasing in length and secondary is increasing in thickness. A reminder that this video focused only on primary growth. We talked about the 4 main types of primary vascular tissue, or vascular tissue resulting from primary growth, which are metaxylem, protoxylem, metaphyloem, and protophyloem. We also talked about a new kind of tissue called meristem, which is unspecialized and constantly dividing. We talked about the 4 main functions of all roots, and we covered some types of specialized roots. Remember that you will often see a combination of specialized and unspecialized roots working together in nature. We talked about the 2 main young tree root systems, taproot and fibrous root, and how they don't always line up with dicots and monocots respectively from the generalization given to angiosperms as a whole. We also talked about the 3 main mature root systems, lateral, oblique, and taproot, and how root growth will sometimes not fit neatly into any of these 3 categories because it is opportunistic. We built a cross-section of a mature root, including the root hairs and root epidermis, the cortex and endodermis, and the stele, which includes the pericycle, conjunctive tissue, and vascular tissue. During this, we covered the differences in the structure of the stele between monocots and dicots, including being protostelic versus siphonostelic, as well as monocot steles containing piths. We talked about Casparian strips in the endodermis that prevent apoplastic transport and force symplastic transport so the root can control what enters the vascular tissue and how this creates root pressure and can lead to guttation. We talked about the root tip structure with the 4 main parts being the region of cell division, the region of elongation, the region of maturation, and the root tip. Remember that despite the visual provided in this lecture, the regions often overlap and are not clearly defined, as they merely represent the activity the majority of cells are doing in a given region. We talked about the mutualistic symbiotic relationships called mycorrhizae, where fungus in the soil helps trees absorb nutrients like phosphorus better. We also talked about another mutualistic symbiotic relationship that occurs between tree roots and soil bacteria, which allows the tree to absorb nitrogen that would not normally be available for them to use thanks to nitrogen-fixing bacteria. That's all, and thank you for watching.